شكرا.
Bismillah ar-Rahman ar-Rahim. It's a quite pleasure to start the second session today by introducing my dear colleague, Professor Dr. Sama Ali, one of the eminent professors in biochemistry and member of the Egyptian National Committee of Biochemistry and Molecular Biology. And he is also head of a lab in Children's Cancer Hospital 57357. Welcome, Dr. Sameh. Please, the floor is yours. Of course, I have to welcome my dear Professor Dr. Belbis here at the stage as well as a chairman of the session.
Thank you so much, Dr. Mohammed, and thanks to the committee that I used to be a member of. I think they are all of them are angry of me because I'm not really attending regularly, but I'm hoping that they will forgive me. Dr. Hassan and the rest of the committee, thank you so much for having me and for inviting me to be part of this exciting event. The subject that I'm gonna be talking with you about is regarding COVID-19, and probably many questions would arise when you see the title. The first question is usually, why in the world are you interested in COVID-19 while you're working in a pediatric oncology hospital? The answer to this question is that when we started this effort, it was to explore the interaction between COVID-19 and pediatric oncology patients. But we drifted from this goal due to some technical issues, and I'll tell you our journey, just a very small account of our journey regarding the understanding of the pathophysiology of COVID-19. Is COVID-19 still important? I think yes. I think it's here to stay. As a conclusion, it's here to stay, and probably we have it with us here in this podium. Not to scare you off, but this is actually a well-established thing. If you look at the most recent statistics regarding coronavirus cases, you'll see that it's now approaching 7 million cases, 7 million confirmed deaths from COVID-19, more than 95 million cases of confirmed cases. Also, the active cases right now, while I'm talking with you, is over 21 million cases. This is, of course, surprising because everyone now is thinking that we are out of this catastrophe, whereas actually it's a kind of global Sisyphus. It goes down and then it comes back again and bounces back again and so on. So understanding the pathophysiology of COVID, especially that there are some implications that's called now the long-term sequelae of COVID-19, which is a long-term or a long COVID. And all of us who actually got the infection are suffering from, and are plenty of things that also led this pandemic to move quickly since its discovery back in 2019 to be number 9th. Now we are actually number 7th in terms of the biggest reported and recorded public health concern and pandemics ever recorded in the history of human beings. So as you all know, the target of COVID-19 is plenty. I mean, it just targets everywhere. The common wisdom is telling us that we would probably suffer from what's so-called cytokine storm and also organ failure, but the mortality is actually was quite high during the peak of the pandemic. We have been interested in answering a specific question. In a relatively homogeneous cohort of patients, critically ill patients that are hospitalized in the ICU, why some of them end up dying while others survive the severe symptoms, although it's a relatively homogeneous cohort of patients. So far, we have been able to publish two manuscripts. The first one is regarding the albumin damage as a surrogate marker for COVID-19 mortality, and it was published in eLife. We have been able to detect some damage in the human serum albumin, and we were able to obtain biophysical parameters that could predict the mortality of COVID-19 patients, critically ill COVID-19 patients, within 2 weeks of collecting the blood sample with a fidelity exceeding 95% accuracy. Then we move to understand the hypercoagulability in the blood of COVID-19 patients. And we focused in a paper that was published very recently in the Frontiers in Immunology on the activation of platelets and the tendency of hyper-aggregability with other blood cells, including the arrowheads of the innate immune system, including neutrophils and other cells like macrophages and monocytes. We actually could find that in patients with a bad mortality outcome, there is increased level of what's so-called giant platelets, and these platelets are hyperactivated, leading to hypercoagulability with neutrophils and increased levels of oxidative stress and also hypercoagulability. So the story that I'm going to tell you about today is now we are starting to understand that neutrophils are really playing a very critical role in the pathophysiology of COVID-19, specifically in critically ill patients. The current story is regarding something that's quite elusive, as you will see from the data that we're going to show now, is regarding the hypoxemia, which is a very common clinical observation manifestation of COVID-19 infection. So hypoxemia, and also the lactic acidosis. Lactic acidosis is increased level of lactic acid and increased level of blood acidosis. And this is a characteristic also, there are some sporadic assays that are reporting the increased level of lactic acid in the blood of COVID-19 patients. Again, neutrophilia is one of the most consistent clinical feature of severe COVID-19 infections, and it actually is reported to increase up to 70% of all patients. Sometimes they are more than 80% of the whole blood cell count, excluding RBCs. So the present study is trying to shed some light on if there are some interconnection between these parameters. It's a very tricky, kind of ambitious goal, but I will let you know our progress in this efforts. So the first thing that we asked ourselves is whether metabolic rearrangement, metabolic rewiring of neutrophils can actually lead to the increased production of lactic acid. It's a very natural kind of hypothesis. And to study this, we collected blood samples from a total of about 102 patients, hospitalized patients, and we divided them into a prospective outcome, mortality outcome, and compared them with control subjects, about 31 donors. So the study is actually is to try to see whether neutrophils are systematically increased in these patients. And as you can see, there is a dramatic increase in neutrophil count in severe patients, which essentially also increase more in the patients. These are the non-survivors, and the blue one usually will be for survivors. The next thing that we studied is the relationship between mortality and between neutrophil count. So if you analyze mortality by ROC analysis and then you use a threshold value as a prognostic marker for mortality, you end up having some stratification based on the increased level of neutrophil counts, and you will see that there is statistically significant survival analysis leading to using neutrophil as a successful prognostic factor in these patients. Okay. So we asked ourselves why there are increased level of neutrophils. Neutrophils are very dynamic population of blood cells that are part of innate immunity. We asked, of course, there are some always dispatch of these neutrophils based on the presence of any infection, but usually if there are no infections, these cells are highly apoptotic. So the turnover of these cells are very high. So usually you will have more than 50% of them are positive for apoptotic markers. So what we did is we explored whether the COVID-19 severity and mortality affected the level of mortality, or the level of apoptosis in these cells. And as you can see, there is some sort of silencing for apoptotic surface markers, as you can see from this analysis by flow cytometry. Then we moved to analyze the expression level by microscopy, by fluorescence microscopy. And as you can see, these data confirms that the expression of annexin 5 on the surface of these neutrophils, isolated, purified neutrophils, are far lower than in control, which is an indication that the apoptotic mechanisms are impaired in these cells. We went over and did some transcriptomic analysis of purified neutrophils. And to make a long story short, what we found is that microRNAs that are linked with genes controlling apoptotic pathways are actually downregulated. So the conclusion of this part of the talk is that in these neutrophils, and specifically in non-survivors, the neutrophils are exhibiting a sort of impaired apoptotic markers. So we went forward and we studied the level and the relationship between this increased level of neutrophils with plasma lactate level and also with plasma pH. And as you can see, although the scatter is very high, but you can see from this presentation that there are some statistically significant correlation between the plasma lactate and between the neutrophils, which is implicating a neutrophil as a potential source contributing to the level of lactate in the blood. Again, we monitored the pH of these patients, and as you can see, these are all color-coded. The red pellets are for non-survivors, and the blue ones are for survivors. And you can see that there are some tendency of lower pH at higher neutrophil count. Again, we also tried to connect this with the oxygen carrying capacity in these patients. And what we did is actually we analyzed the relationship between hemoglobin oxygen saturation and between the oxygen pressure that is required to reach the 100% saturation of these hemoglobin. And you can see that the curves here are showing some sort of right shift for the curves of the non-survivors. This indicates that you would require higher oxygen pressure to reach hemoglobin saturation, which also, again, an indication of an impaired sort of oxygen carrying capacity. And this points at an important role for neutrophils in this kind of phenomena. Then we attempted to look at the pH value, and we see that for the non-survivors, the pH are significantly lower in the blood relative to survivors. Again, we analyzed the plasma level of lactate in frozen samples from these patients, and we can see, again, an increased level of the non-survivor levels of lactate in these patients. Then we wanted to really to confirm, we started with the hypothesis that metabolic rewiring in neutrophils probably are responsible for increased level of lactate production, but to our surprise, we actually didn't see any metabolic rewiring. We didn't see any increased neutrophil production of lactate on the individual level of neutrophils. But when you consider the fact of neutrophilia or the increased level of neutrophils in the blood, you then end up getting some information that probably neutrophils are contributing to the lactic acidosis. Then we moved to study the lung damage and tried to correlate lung damage with survival outcome. And you can see here from a measure of lung damage in some sub-cohort of these patients that non-survivors are exhibiting much extensive lung damage. And when we did analysis, survival analysis based on this lung damage, we could obtain also statistically significant predictor of the survivor based on the CT lung damage score. So the conclusions that we presented a kind of evidence substantiating the role of neutrophilia in hyperlactatemia, in blood acidosis, and in impaired oxygen transport function in non-survivors. We found no metabolic shifts in individual neutrophils. However, when we took into consideration the increased level or the increased number, the dramatic increased number of neutrophil count, we actually factorized and we ended up getting an information that probably neutrophilia are contributing to the blood acidosis. So we think that we add a new dimension to the detrimental role of neutrophilia, and most of our results are now pointing at neutrophils or increased level of neutrophils during COVID-19 pathophysiology as a primary cause of the mortality outcome. I'd like to thank all of my group members who contributed to this work. Without them, I wouldn't be here talking about it. I also thank the hospital because this work is funded completely by the Egypt Cancer Network, Canada and America, and also by the hospital, the 57357 hospital, and the colleagues that contributed to this from Qasr Al-Aini, and also colleagues in the hospital who did the analysis of transcriptomics, and my group members who actively worked on this in a very under very challenging environment. And I thank you very much for your kind attention.
Thank you, Professor Sameh, for this very enjoyable lecture. Now, Professor Lual will present the next speaker.
Thank you, Prof. Our next speaker is Professor Dalia Saleh. Yes. Professor Dalia Saleh is Professor of Molecular Pharmacology from National Research Center, Cairo. Go ahead, Professor Dalia, you have 15 minutes. Thank you.
Thank you. I would like to thank Dr. Azazi and the National Committee of Biochemistry for inviting me here today. My presentation today is entitled Uncovering the Mysteries of Peripheral Diabetic Neuropathy with a Comprehensive Exploration, Pathogenesis, Diagnosis, and Management, presented by me, Dalia Saleh, Professor of Pharmacology, National Research Center. The outline of my presentation today includes an introduction, classification, symptoms on diabetic peripheral neuropathy, how to diagnose, the pathogenesis, and the management of how to manage the diabetic peripheral neuropathy, and finally, conclusion and future recommendation. First, I would like to give a brief introduction. Diabetic peripheral neuropathy is a chronic complication that affects more than 50% of diabetic patients. However, half of these patients suffer from painful sensations, which is commonly harsh. It leads to depression, impairment of quality of life, lead to sleep disorders, as well as the painful sensation. These patients suffer from major clinical concerns, example, leg ambulation, which may lead finally to death. Neuropathy is a nerve damage that begins in the distal lower limbs. It appears as numbness, tingling, discomfort, and weakness. This is due to mainly degeneration of the axons, ischemia, reduced Schwann cell axon transport, and in more severe cases, demyelination. How to classify diabetic peripheral neuropathy? Diabetic peripheral neuropathy is classified to many classes, the most important of which is the distal systemic polyneuropathy, as it comprises about 70 to 90% of diabetic peripheral neuropathic patients. These patients suffer from shooting pain, burning and tingling numbness, impaired balance, and foot deformities. How to diagnose peripheral diabetic neuropathy? First, we will take a history on the patient, assess the symptoms, then we will make some diagnostic or perform some diagnostic tests, example, as the electromyography to measure the electric discharges in the muscle, nerve conduction studies as to measure how the nerve fibers extremity conduct the signal.